Hi, good morning, good afternoon, good evening. You're welcome to class today. My name is Sam. I'm going to be your biology teacher today. And in this class, we'll be looking at A-level topic, which is actually chapter 6 in your A-level syllabus, which is nucleic acid and protein synthesis. Okay. All right. In the course of this, we'll be looking at for two parts. I actually grouped it into two parts as my objective. The part one, we're talking about nucleic acid. Then under this part one, we have the structure. We'll talk about the structure of the nucleotide. We look at the RNA and the DNA. And then we look at replication, how this DNA replicates. And then in part two, we look at the protein synthesis. So we look at the codons, we look at transcription, translation, we look at the gene mutation, and then we also look at sickle cell anemia. Okay, let's look at the introduction to genetics, as in what this genetics is all about, why we study genetics. Look at the characteristics. We have two things majorly that this gene do what they are known for. One is the ability to carry instruction or information. DNA, for example, is referred to as the blueprint, the blueprint of who you are. So what you see on the physical has a lot to do with the gene you carry inside of you. Okay, so if you are dark, that means you have a gene for darkness, for dark. I said darkness. Sorry about that. If you are fair, you have a gene for fair. If you are tall, you have a gene for tall, and so on and so forth. Okay. Next one is ability to be copied. Now, these genes can be copied during reproduction. That is meiosis. Your parent gene, your dad and your mom actually give half of their gene will be copied into you. So that's why you look like. Okay. All right. Then also we'll talk about the stock. This DNA as a whole is discovered by these two gentlemen. The one we call the James and the Francis. Now we know about Gregor Mendel. Now, Gregor Mendel was actually the first that discovered the differences, the appearances, the differences in the characteristics of organisms. We read about him, you know that he's a scientist. He worked on pea plants. He has a garden, and then he discovered that some of the plants, some of the pea plants here, some are tall, some are short, some are yellow, some are green, some are round, some are wrinkled, and so on and so forth. So these are the observations he made that created that curiosity in him that made himself start studying. But he has no information about what gene or DNA is all about at that moment. Okay, so that's why today is being um, credited the father of genetics, because he is the one that started the trend. Then subsequently, this young lady called Rosalind Franklin took up the whole process and started doing her research. Now, I can't give you the full information. You can research about them later on. You'll get to know more about them. But she started this research, started working on the DNA before these two gentlemen came. And why we have used the word controversial is because of the fact that these two gentlemen actually worked on what Rosalind had, but they didn't give her the credit. So they are regarded to be that they stole Rosalind's information. So they're supposed to give Rosalind give her credit because she was the one that started researching on this actually, but they didn't. So that's why the thing created this controversy among them. Like I said, you can read that, you can go do your research and find out more about these three gentlemen. Okay, so let's look at nucleic acid. Now, when you say nucleic acid, first thing that comes to your mind is the nucleus, the acid of the nucleus, because the nucleus is where this DNA is. Okay, so these genetic materials are actually in the nucleus. So I think that's where they got the name nucleic acid. And we know that we know when we talk about nucleic acid, we are also referring to both prokaryotes and eukaryotes, because we said every living organism, not just cells now, every living organisms possesses this nucleic acid. Okay, so prokaryotes, they don't have nucleic membrane, but they have the components. Okay, so I believe you've done that before in your previous topics. Then we'll talk about the monomer of this nucleic acid. We refer to it as nucleotides. So monomer of this thing is called nucleotide. Now, then also there's also the one called polymer. Now, polymer is referred to as polynucleotides. Now, we don't usually use the word polynucleotides. We rather use the word DNA or RNA because these polynucleotides are of two types. So we have the DNA and we have the RNA. So DNA is a combination of multiple monomers. Same thing with RNA, combination of multiple monomers. But there's a slight difference between the DNA and the RNA, which we're going to talk about in the next subsequently. All right, so we'll talk about the bond that holds these monomers together. The bonds between these two monomers that brings about formation of this dinucleotide or polynucleotide. This bond referred to it as phosphodiester bond. So this bond is called phosphodiester bond. Then also there's also a bond that holds the complementary base pairs. Now we'll talk about these complementary base pairs, but I'm going to do just a brief introduction of what it's all about. Now in DNA or in RNA, each of them they carries a 4 base pairs. Now this complementary base pair is also referred to as the nitrogen base. Now this nitrogen base there are 4. We have the uracil which is found only in RNA. Then we have the adenine found in DNA and RNA as well. We have the thymine, which is also found, which is only found in DNA. We have the cytosine, which is found in both. And we have the talked about guanine, which is also found in both. Okay, so these are the nitrogen base. Now, these nitrogen base occur in pairs. Okay, so adenine can only pair with thymine or with uracil. We'll talk about that later. Then while cytosine can only pair with guanine. Okay, so when these two nitrogen bases pairs, now there's a bond that exists between them. The bond that holds them together is what we call the hydrogen bond. So that's what hydrogen bond is. So we'll talk about diester bond that holds the nucleotides, and then we'll talk about the hydrogen bonds that holds the nitrogen bases. Okay. Next, we'll look at the nucleotides. Now, nucleotides consist of 3 components, which we've mentioned some of them just now. So we talked about the nitrogen base, which I mentioned 4 of them, which including uracil. So we have again the adenine, the thymine, the cytosine, the guanine, and then the uracil, which is only found in RNA. Okay. Now, these nitrogen base we just mentioned, they are grouped into 2 based on the number of rings they carry. Based on the number of rings. Now we call the purines and the pyrimidine bases. Okay, so we'll look at it in the next chapter, in the next page. Now the rings, some they have just one single ring, while others carries a double ring. Okay, so that's all referred to them as purines and the pyrimidine bases. Then we also have the pentose sugar. Now this pentose sugar, although we have 3 sugars. We have the pentose sugar, we have the triose sugar, and we have the hexose sugar. So here we're dealing with the pentose sugar, which comprises of 5 carbon sugar. Okay, now this 5 carbon sugar can either be deoxyribose or ribose. Now deoxyribose meaning that it has, I'll use the word short 1 of oxygen. Short 1 of oxygen or lesser oxygen, while the ribose has a full oxygen. Okay, so that's why I use the word deoxy. So that's what differentiates a DNA from the RNA. Deoxyribonucleic acid, and the other one is ribonucleic acid. Okay, so that's deoxy, reduced oxygen. Okay, then we also have the phosphate group. Now this phosphate group is negatively charged. That is why DNA is a negatively charged molecule. So take note of that. Take note of that. Now, if you look at this structure here, this is a structure of a DNA. You can see this H here. There's no O. This is the pentose sugar. This is the pentose sugar, which is the deoxyribose. So there's no oxygen attached to this hydrogen. But if you look at the RNA, you can see there's OH here. So that's what makes it a ribose sugar. Remember, I said short 1 for deoxy and normal for ribose. Okay. All right, so this is the nitrogen base. The acronym here stands for adenine, cytosine, guanine, uracil. That's for RNA. Then come to DNA, you have adenine, cytosine, guanine, and thymine for DNA. Now here is a phosphate group, which we said is negatively charged. Okay, so that's what we have here, and this is ribose sugar. Okay. Okay, next we'll look at this nitrogen base. Look at these nitrogen bases. Now they are, like I told you before, they are divided into two. The four nitrogen bases are divided into two based on the number of rings they have. So purine has two rings. So that's why they use we use the word pure as gold. Pure there is referring to purine, as is referring to adenine, while G gold there, the G in the gold is referring to guanine. So pure as gold. So anytime you're asked to mention the nitrogen bases, the purine nitrogen bases, you remember the acronym pure as gold. So adenine and guanine. Then the next one is pyrimidine. Pyrimidine has one ring only. Now the reason, the essence of this ring is to bring about the balance so that both of them will have same density. Now look at the structure here. This is adenine. Adenine has two rings, so adenine can only be complementary to thymine. Adenine can only be complementary to thymine. So that is the balancing now. So two rings, one ring, that's making a total of four rings. Now look at next one, cytosine and guanine. You can see cytosine is one ring, which is a pyrimidine, while guanine is a double ring, which is a purine. So the two of them as well. So 1, 2, 3 rings, 1, 2, 3 rings. So that's how they work. So remember pure as gold. Then you have the pyrimidine, you have the cytosine, the uracil we mentioned before. That's for DNA only. Sorry, for RNA only. And then the thymine for DNA only. So where there is uracil, there is no thymine. Where there's thymine, there is no uracil. So in RNA molecule, what you're going to see is adenine, guanine, cytosine, and uracil. So there's no thymine. So take note of that. All right. So then we'll talk about the complementary pairs. Now, complementary pairs, we talked about that before. The ones that complement, the one that can go. Now, pairing of bases is very precise. It's very precise. Now, if you look at this first one, look at the T and A, the thymine and adenine. You can see the number of bond. You can see first bond, second bond. There's no third one. So they have two bonds that's holding them. Then look at this one. They have three. See the first one, second one, and the third one. Okay, I think that's also part of one of the things that makes them complementary. So they are precise. Now, purine always bind with pyrimidine in order to balance. You know, we talked about the rings. So in order to balance density, so that's why they bind like that. So DNA molecule has same weight throughout. So that's what the complementary is all about. We'll look at the structure of RNA and DNA, although we've talked about most of it in a subsequent part. So RNA stands for ribonucleic acid. The ribo is referring to sugar. The is ribo sugar, the pentose sugar. The nucleic acid referring to acid of the nucleus. Now they are single-stranded. Now they form what we call single helix. Sorry, single helix. Now you can see the helix, single, unlike their DNA. So DNA has double helix, while this one has a single helix. And then on this helix, you will see the nitrogen base. And because it's RNA, that's why I have a uracil instead of the thymine. Okay, so again, because it's a ribose sugar, remember what I said for deoxyribose, that is referring to short one. That is one oxygen is missing. But in ribose, we have one oxygen atom more than deoxyribose. Okay. All right. So they also have a phosphate group. Now we're going to talk about this helix this helix that is referred to as a backbone. We're going to talk about the components of this backbone next. Okay. So that's for RNA. Now, take note of this. We always talk about the ATP, the adenosine triphosphate. Now, if you look at ATP, you can see the same. It has a adenine group, so one adenine, then three phosphates, one adenine and three phosphate. Now, this adenosine is referring to adenine and ribose. That's why I call it adenosine. Okay. Now, this is the three phosphate group. Remember the phosphate group that are negatively charged. for RNA and DNA. So these are the 3 phosphate group. This is the 1 ribose sugar, and here is the adenine. Remember, adenine is a purine. Adenine is a purine. Remember, pure as gold. So it's a purine. So that's why I have 2 rings. Okay. Now, if you come to this ribose sugar, even if they didn't give you the name of the name, what you should know here is the fact that there is oxygen attached to this hydrogen here. Remember, we said ribose, they have 1 oxygen more than deoxyribose. So by looking at this is what gives you the information that this is a ribose sugar, not a deoxyribose. Okay. Okay, so that's the structure of a DNA. Remember, a DNA is a RNA nucleotide. Remember, all of them are all nucleotide. But when this nucleotide starts if I say it's an RNA nucleotide, it means that it's a monomer. So by the time it starts combining, it forms the polymer. Okay.